Okay.
Hello, Dhaval.
Yes, I am okay now.
Dhaval, you can start your seminar. We already can hear you now.
Dhaval.
Okay. Okay. Thank you. So again, a very good afternoon to all of you, and thank you for attending this webinar. I will discuss today the journey of innovative mass spectrometer, which we have developed through focused innovations to make it more accessible for everyone. So if you are familiar with mass spec, is actually a very flexible and versatile technique that can be used for different analytical challenges across different industries and application areas. For example, if you have a sample of biological origin, you may be focusing on discovering biomarkers or developing new macromolecules as a new therapeutics drugs. It can be focused on detecting contaminants or in the environment or developing chemical materials towards the next generation of, for example, plastic and fuel materials. And we can also use for research and development ranging from food safety, food research, natural products, and many different applications. However, historically, there have been a lot of challenges for people to deploy mass spectrometer in their laboratory, as I have listed here, ranging from its routine use, cost, to perceived complexity of its use, and also maintenance. So how we at Waters, being a specialty measurement company, overcome these challenges, that's what I'm going to cover in today's presentation. So we can take an example of our day-to-day life electronics items. So as the technology gets smaller, easier to use, and more affordable, it is more accessible. So then we have from small group of users to well-funded users to millions of users and even billions. So if you take an example of mobile phones, a laptop, a camera, and nowadays we are talking about wearable devices. So because of this revolution, we have a lot of people are able to use this technology. So mass spectrometry is also following a similar trend. So what you can see here in the past, where we have one big room required to host a mass spectrometer. And from there, late in 2002, you can see a mass spectrometer on which there was work done to develop electrospray ionization, which got a Nobel Prize at that time. And from that, we come out and launch a new small mass detector a few years ago, which is like a stackable mass spec on top of your LC stack or below your LC stack. So we revolutionized by launching this small miniature compact mass spectrometer, empowering a lot of chromatographers around the world to harness the power of mass spectrometric data. And by adding this mass detection to their workflow, you can get a lot of benefits. For example, you get ability to detect UV transparent compounds. That gives you much more specificity, particularly in case of finished products where you might have interference from co-eluting compounds or other compound. You get a better sensitivity of this particular instrument, so that you can even measure low-level contaminant and analyze within your sample. So when we commercialized this product, a lot of people adopted it, and now we have thousands of users worldwide routinely using this mass spectrometer. However, when we talk to those users, we realize that there is an increasing demand to go beyond a normal mass spectrometer, where people want to utilize high resolution mass spectrometer routinely for their analytical challenges. For example, if you're looking to profile compound in your natural products, trying to identify compound in your food products, doing some impurity analysis, and so on. And high resolution mass spectrometry is very powerful technique because it provides you elemental composition so that you can identify unknown and unexpected components in your sample. It is more specific so that you can get more information, more confidence in your data. It provides you a more accurate information on the structural level so that you have more confidence in identifying the compounds of your interest. So what does it mean by accurate mass? So we can take this comparison of this normal balance versus a micro balance. So if you measure something with normal balance, you just get mass to 1 decimal point. And when you measure something using micro balance, we can get up to 4 decimal point. So high resolution mass spectrometry, it gives you accurate mass up to 4 decimal points. And by using this very specific mass to charge values, we can differentiate compounds with very similar masses, as we can see here. So if I give one example, if you look at these 3 molecules, amitriptyline, venlafaxine, and entecavir. So these are 3 drugs molecule having very similar masses. So if you use a normal mass spectrometer, which are of low resolution, we may not be able to differentiate these 3 compounds. But when we use high resolution mass spectrometry, by using their mass up to decimal points, we can confidently separate these 3 molecules. So this is the power of a high resolution mass spectrometer. And on top of that, if you have some unknown peak, it would generate chemical formula so that you can identify your unknown molecule. How about biopharmaceuticals? So biopharmaceuticals are much more complex molecule. As we can see, the molecular weight ranges from more than 100 kilodalton to all the way 200 kilodalton and even more. And because of this large molecular weight, small changes in their modification, we will not be able to measure accurately using a normal mass spectrometer. We need very specific high resolution mass spectrometer, which can allow you to measure small changes in molecule. And this could be a critical for the safety and efficacy of this molecule. So to give you perspective, this is a paper which was published a few years ago by authors from FDA. They analyzed about 80 applications submitted for biologic licenses in US. And they mentioned that out of 80 applications, 79 of them have used mass spectrometry for their protein characterization or impurity characterization. So this got to show that the mass spectrometer plays an important role in drug discovery analysis. And we did this survey where people say that they want to use high resolution mass spectrometry routinely for their work. However, there are challenges that I mentioned earlier. And if we can make this mass spectrometer more accessible, they would be more than happy to use them for routine applications. So how we make this more accessible? So from our experience using the QDA detector that I described earlier, we use a similar approach to make a high resolution mass spectrometer, which traditionally looks like this big instrument, very difficult to use. You need a certain level of expertise, to something which is very compact. It actually is smaller than your liquid chromatography spec system. If we zoom into the dimension, you can see it's very compact, high resolution mass spectrometer specifically designed for people who do not have any mass spectrometer experience. So the goal behind this mass detector, we name it as Acuity RDA detector, which stands for resolution Dalton, because although it's a small compact mass spec, actually it gives you high resolution accurate mass data. It's of small footprint, which is ideal for many labs, so that you can easily put this instrument in your lab. And on top of that, if you need to do some compliant ready analysis, for example, you need to comply to GMP, GLP, or let's say 21 CFR Part 11 compliance, this instrument with the software is capable of doing so. So how does this instrument looks like in actual physical dimension? So I will play a very short video here. So you can see this is an LC stack, and this is the Acuity RDA mass detector, which gives you high resolution mass spec data. And you can see actually it is smaller than your HPLC or LC stack. And we call it as a smart MS system because there are certain features which we implemented in this system, and probably this is the first smart MS enabled solution, as we term it. So it has one button startup, so that you can easily and quickly get onto this machine to do your experiment. it does its automated setup. So if you're doing a high resolution mass spec analysis, there are a lot of setups or tuning required in traditional instrument. However, with this instrument, you can just do it automatically by just one click. And we have a lot of intuitive user interface and diagnostic inbuilt into the system to make it what we call as a smart mass spec. So I will show you some of the features, what I mean by smart MS. So on this instrument on the front panel, we have this interactive panel with this push button. So this user just need to push this button to get it started. So this is to ensure that all your analyst has a similar startup and shutdown procedure so that you don't encounter any errors or difficulty in using the system. What about tuning the system or setting up? User do not need to do any tuning or setup at all. You just come to this page, select the mass range of your interest. For example, this instrument is capable of achieving or analyzing molecule up to 7,000 Dalton, which is quite high compared to what you can find in other mass spectrometer system. And by just clicking this start button, the machine would automatically able to set it up by doing the required setup. And although this is very small benchtop instrument, it is equipped with high resolution and high stability top mass analyzer so that you can get reproducible data with a lot of accuracy all the time. I mentioned about the intelligent diagnostic system. So again, on this front panel, actually there are different fluidics and interactive diagram of the instruments there. So each of this specific diagram or figure here would indicate the health of the instrument. So even without touching the instrument, you will clearly know whether the instrument is ready to run, whether it requires some setup, or whether there is any problem and you need to call the service engineer to fix it. So now we are implementing those smart features that user can just look at from distance by looking at the instrument without even touching the instrument. And I'm going to show you some example data, but the instrument method is very easy to set up. We have a full scan MS as well as full scan MSMS acquisition data modes. And there is this acquisition mode, what we call as data independent mode, where user can collect the MS and MSMS information for all the possible ionizable analytes within a single run. So this is a small but powerful instrument. So let's look at some example data. I will start with small molecule applications. So although people have started using high resolution mass spectrometry, but historically there is challenge to do the data analysis for high resolution mass spectrometer, because since we get more specific, more information, so more information, that means you need to spend more time to analyze the data. So when we launched this system, we also looked into how we can streamline the results generation from sample injection to the analysis and getting the results. So we did it by implementing a dedicated workflow. So this solution has Streamline Workflow, which enables you to generate results. I will show one example later. And we have built in a lot of tools which are required to do the identification and elucidation of the compounds, including scientific library covering all range of applications so that you can do the screening and profiling of your sample. We have a lot of online as well as inbuilt elucidation tools. So let's say if you come across some unknown peak or unexpected peak, how we can identify what it is. We have all the tools available within the software. And this solution also has a functionality so that you can do automatic report generation. For example, you set up your sample. After acquiring the data, it will process the data and will automatically generate report by itself. So you just need to look at the results. So we have done a lot of automation so that it can do much more efficiently and much more productivity. Okay, so the idea behind this software is to let's ensure that people can quickly learn and easily master to do their routine work. I mentioned about elucidation tool set. So this is one example of data. Let's say we came across this mass. We were not able to identify it from the libraries that we have. So we use this elucidation tool set where user need to click this button. The software or the solution would generate formula for this compound. It would search against ChemSpider databases. It would rank all the possible matches based on the fragmentation analysis. So again, it will also do the MSMS interpretation of the data to give you out of those possible hits, which possibility is likely identity of compound. So again, we reduce a lot of requirement on expertise and understanding of mass spectrometry so that anyone who with minimal knowledge can get onto the system and generate the results that they want. Okay. So the first example I have is of honey, but I mentioned earlier, we are using dedicated workflows covering a wide range of applications. So we already have this workflow existing for our other high resolution mass spectrometer based solutions. So we just use that existing workflows and repurpose into this solution so that you can use it immediately to do your analysis. So let's look at one quick example of honey. So there is a lot of interest in the people to know whether it's real honey or factory fake, because every honey is a unique complex matrix made up of plant secondary metabolites ranging from flavonoids, phenolics, sugars, and additionally, there are natural compounds from bees as well. And the fingerprint of particular honey sample would depend on the region, forage, as well as the biological properties. So we did this study by collecting samples, commercial sample, as well as authentic honey samples. And we did ultrasonic extraction, and using this activity RDA UPLC analysis, coupled with multivariate statistical analysis, we were able to classify these 4 types of honey as well as the commercial products into a different category. So this is a very characteristic usage, how we can use this to do the profiling and to discriminate your samples, be it food, natural products, or any other samples that you want to do. You can do it by using this non-targeted screening application. The other application I want to discuss is the study that we did with our collaborator in France, who are CRO companies. And in this case, they wanted to do impurity analysis of a generic molecule. So their objective was to identify and confirm the 4 impurities that they know previously. And then they wanted to do the finished product stability study profiling, as well as trying to get idea of potential origin of this impurity from their stability studies. The high resolution mass spectrometer is a powerful tool if you're doing impurity characterization, because sometimes there is subtle change. For example, changing, let's say, a methyl group changing to ketone group. The small change in decimal points will not be measured by using a traditional nominal mass spectrometer. You need a high resolution mass spectrometer to measure such subtle change. And that's where there is also increasing interest for people to use high resolution mass spectrometer to identify impurities, because it can give you a lot of other information. For example, it can tell you the source of contamination. You can identify potential dimers. You can also characterize something that is unexpected, and so on. So for this sample, these are the representative chromatogram of those 4 impurities, as we can see, based on UV and mass spec profile. And within the software, we have inbuilt transformations that are available for users, so more than 100. You can also customize your own transformation. So by using this transformation, we identified these 4 impurities and confirm by looking at their chemical formula and these excellent mass accuracy values that we derived from this compact TOF mass spec. So we confirm all the 4 impurities based on their accurate mass and the mass accuracy values. So then the next question is to analyze the API product, which was subjected to 18 months stability studies. So upon those accelerated stability study, they detected 3 peaks in their sample, as we can see here in this chromatogram. So the objective was to identify what are those 3 peaks. Okay. So the first, we confirm that the prominent peak that we see is of API. So we can see this is a very clear precursor ion mass, and these are all the daughter ion mass. So as I mentioned earlier, the instrument is also capable of doing a simultaneous full scan MS and MSMS acquisition or data independent acquisition. So within one injection, we get full scan MS and MSMS data. The software will also interpret the MSMS data for you. As you can see, it will interpret and tell you that from this structure, by breaking of these different bonds, we can get this kind of fragmentation pattern. So again, user do not need a knowledge of mass spectrometer. The software is designed to do it for you. And then we confirm, again, similar approach by using a chemical formula is named. So this was a bisoprolol drug, which confirms with the API that they use. Then we started to zoom into each of these impurities. So we can see here the one retention of 6.8. Similar approach by looking at precursor mass and daughter ion mass, we identify it as impurity K. And again, this was there with our library based on the impurities that we run earlier. We confirm that this is this particular impurity. Likewise, for the impurity is 6.60, this impurity does not exist in the library. So we look at the fragmentation or the mass spec data carefully. Then we can see this characteristic isotopic fragmentation suggesting a chlorinated component. So based on the chemical synthesis knowledge from pharma physics, they realized that this compound is unlikely related to their API or the synthetic molecules that they use. Okay, so then this was ruled out. Then the third impurity, again, it was very low mass, about 100, was not matching again with any of our library entries. So we use those elucidation tool set to identify what it is. Then by doing those elucidation tool set, the software proposed 4 different identities from based on the mass and the structure. So these are the 4: benzaldehyde, tropone, phenomethone, and the third one. So again, based on the synthetic knowledge, they confirm that out of these 4 possible entries, only 2, as I have highlighted in these green color boxes, are the likely identity of this impurity molecules. So by using this compact mass spectrometer, they were able to confirm the impurities, identify the potential impurities that could be formed as a part of the stability studies. And this is the slide that I borrowed from them. They say that by using Equity RDA, they were able to save time. They were able to generate more compelling data so that it is more convincing for regulatory authorities. And being a CRO companies, by implementing this high resolution mass spectrometer, they were able to get much more contracts based on the data that they presented to their customers. And this is the quote from the director, that it reminds them of ease of use with the power of high resolution mass spectrometer. So again, this is a really very powerful tool for your small molecule analysis, regardless of your sample or application. How about large molecule? So although it's a small, compact TOF detector, it can perform equally well for your large molecule. So in next 15 to 20 minutes, I'll show the large molecule data as well, with the example of biologics and some data on COVID-19 as well. So we have a lot of news and updates these days about COVID-19. So there are 3 approaches. Of course, the first one, people want to do more accurately and specifically diagnostics so that they can identify people who are infected with COVID-19. So there have been many tests approved worldwide. I have listed here. And similarly, people also have looked into potential treatments. So FDA, for example, have reviewed about 370 potential treatments. And out of them, they have approved 7. So 5 for emergency use, and 2 treatments which were just approved this month are from Eli Lilly, which are basically a combination of monoclonal antibodies for the COVID-19. And there have been a lot of news about vaccines. So a lot of people or companies have been trying to work on a vaccine. So I will show you very short glimpse of different assays, how we can use this compact mass spectrometer to do this kind of analysis. So let's look at the instrument itself. So for diagnostic test, we are looking at oligonucleotides. I'll tell you why people are interested in oligonucleotides for diagnostic test. For treatments, as I mentioned, you want to look at monoclonal antibodies as well as some small molecule drugs. And for vaccines, there are many different kinds of vaccine, either based on a protein, based on mRNA or DNA-based technique. So what we did, we combined actually that Acuity RDA mass spectrometer into a system solution, and we call it as a Bioaccord solution, because this is a solution which is purposefully designed for biologics and biotherapeutics analysis, which gives you a powerful performance is protein, peptide, and a glycan level, as well as oligonucleotide level. And again, similar to our small molecule, we have dedicated workflows for biologics and biotherapeutics. So actually, this is more than just a mass spectrometer. So we combine 5 components. We have our UPLC. You can use it either UV or fluorescent detector with it. You can use this Acuity RDA compact mass spectrometer. We have dedicated workflows. And on top of that, we have a dedicated application-specific system installation and support, as well as qualification. And then pre-design and optimize chemistry products so that you can do those routine application from the moment you get and install this instrument. Okay, so I'll start with some of the applications. So let's look at the oligonucleotides. So oligonucleotides are usually a short chain of nucleic acid. It can be single strand or double strand. And usually in the past, it was referred to less than 50 nucleotides long. But these days, anything which is chemically synthesized nucleic acid containing molecule is called as oligonucleotides. And there have been many uses from research to diagnostic as well as therapeutics. So how oligos are used in diagnostic kits? So you might have heard this terminology called polymerase chain reaction. So over 30 years, oligonucleotides have made huge impact by facilitating the polymerase chain reaction. So the PCR is like photocopying a page of book for DNA. So but unlike normal photocopying, in PCR, an enzyme called DNA polymerase is used, which has to be bought on paper to make these copies. So the researcher or the companies typically use a short chain oligonucleotides, which are synthesized to have a complementary sequence outside the edge of DNA. So the red color molecules are your oligonucleotides primer. So these are having a complementary sequence to the DNA sequence that you want to amplify. Then by using specific temperature combinations, the target DNA is open and oligonucleotide binds to the complementary target sequence. And then polymerase reactions start copying them. And we have a multiple or even billions of these copies being produced so that you can detect whether a particular pathogen is there or not. In this case, is virus. So one of the key aspects to ensure that this kit works, how you can ensure that the results you are getting are true positive and free from false negative or true negative results. So the success of this test relies on the quality of this oligonucleotide, which are used as a primer. So you need to ensure that you confirm the identity and purity of this oligonucleotides. You separate the impurities from it, and you can confirm the sequence of the primer that is used for this kind of diagnostic test. Okay, so how to check if primers are made correctly, or they are good primers or bad primer? There are certain ways we have to confirm the length so that the length is correct. Then we can confirm the mass and check its purity, whether it has a desired purity to do this amplification or not. We can do this very quickly using this Bioaccord solution. So what you need to do, you just need to key in your oligonucleotides, the masses here. Then the instrument would run them, would separate different oligonucleotide based on their nucleotide bases. It would confirm the mass, as we can see in the spectrum. It would confirm the mass, and it would tell you whether the oligonucleotides that you have is correct and it has desired purity or not. So we can do this kind of analysis for any oligonucleotide molecules, which ranging from 50 nucleotide bases to 100 or even more nucleotide bases. So this is, again, a very straightforward, simple solution to do oligonucleotide purity and mass confirmation. How about monoclonal antibodies? So if you are not familiar with monoclonal antibodies, this is how structurally it is represented. It has two chains, basically light chain and heavy chain, which are joined by disulfide linkages. And on your heavy chain, you will have some glycosidic residues or glycosides attached to it. So when you want to analyze the monoclonal antibodies or biological therapeutic protein, people look at certain cases. For example, they want to look at intact mass. So I want to measure this molecule as intact molecule to see how it looks like and what are the different glycoforms that we can see and what are their relative abundance. And then we go down from this intact molecule to its breakdown product. So we break them into two different chains by doing the reduction and by doing some idealization, we can open up this heavy chain, a light chain, and we can separate them into different subunits. So this whole molecule now broken into 4 different pieces, and we analyze this and we call this as a subunit analysis. So now we are analyzing the subunits of monoclonal antibodies. Again, all this test relies on the accuracy of mass measurement. So we measure the mass value of this side chains to confirm the mass as well as the sequence, as well as any potential modification. For example, here we can see these are the glycan molecules that are attached. And then we can go down further. We break down these molecules into small pieces. We call them as a peptide. So from whole protein or antibody to peptides. And we do this assay we call as peptide mapping assay. This peptide mapping assay is actually one of the regulatory requirements to confirm the sequence of your molecule and to also confirm any post-translation modification that are present in your molecule. And last, if your molecule has a glycan or glycosylated, which a lot of biologics molecules do have, then we release these glycans. These are attached here from your antibody, and then we analyze this released glycan molecules to confirm the glycan profile of this molecule, because all this has impact on safety and efficacy of biological molecules. So it is important to do all these assays. So these are typical assays that people would normally do for their antibodies. So we did this by using our dedicated workflow. So for each of the assays that I mentioned, we have intact mass where you can get data like this. This is the mass spectrum of your molecule. And these are the different glycoform of your antibody present, because antibodies usually have a different proteoform. They never exist in a single form. They will be present in few isoforms. So it's important to know what are the different isoforms and how much of them present in a molecule. Then we can do the subunit by breaking down into different chains. We can look at the peptides to look at the fragments of this molecule, and even we can look at the glycans, as I mentioned earlier, by doing this workflow. So I'm not going into detail, but the solution that I've described is capable of doing all these assays, because I want to, in the last 5 minutes or so, I want to go through the vaccines part, right? So, you know, there are a lot of ways of making a vaccine, but one of the common step they have all the vaccines in common is that representing an epitome, which would generate an immune response in our body, okay? So for that, one of the binding mechanism, or we call as a business end of this viruses, is what we call as S protein or spike protein. So we call spike protein. So this spike protein has a lot of glycans attached to it, or in other words, it has a lot of glycopeptides on it. So it's very critical to measure this glycan profile of your molecule if you're developing a vaccine. So in this case, if you're trying to characterize this S protein of the virus, is important you characterize the glycan profile so that you can develop a proper antigen. So this is just a slide shows the spike protein of COVID-19 virus. We can see here all these molecules are the glycan, which are basically sugars or monosaccharides, units attached by chemical bonds in different positions and different organizations. So we can see it is quite heavily glycosylated. So if you're developing a vaccine using a spike protein as an antigen, is very critical that you know the glycans attached to it very accurately and correctly. We can do so by using this assay, which we call as release glycan assay. So this is just one example of dedicated workflow. We have everything from chemistry kits to running an experiment and generating data. So this is what I mean by doing a streamlined workflow. So for a glycan molecule, because these molecules are not UV absorbing molecules, so you need to do some derivatization to make them fluorescent label. But traditionally, the process of breaking the glycan from antibody, releasing the glycan and labeling them and getting them ready to do the analysis typically takes about a day or few hours. We come out with reagent which can do this thing in about 30 to 40 minutes time with much more ease and simplicity. And you also get much more sensitivity. So we can label this thing very quickly. So again, we can increase the efficiency and productivity by doing quick labeling. And after labeling, we have this dedicated workflow where you can run a ladder. So this ladder is basically a glucose unit, which we use to generate a calibration curve. Then from this calibration curve, we compare our sample chromatogram to derive the glucose unit present for the glycan. And then we can use a library to identify what are these glycans. So we also have a dedicated glycan library that you can use to identify and confirm the identity of glycan in your sample. And typically for this kind of work, you need to ensure that your antigen target is reproducibly glycosylated, so you can run multiple samples and look at the profile. As we can see here, the profile is quite reproducible, meaning your antigen is reproducibly generated for your vaccine candidate. Okay. The last example I have is about the mRNA vaccine. So again, with interest of time, I'm not going into too much detail, but you can contact us later if you are interested in any particular analysis in a detail. So you know, these days Pfizer and Moderna vaccines, they are being mRNA-based vaccines.
Hello, Dhaval, you have 5 minutes more.
Yes, I'm aware. Yeah. Okay. So then this vaccine is produced through these 4 steps. One of the key steps here is doing the capping analysis. So they actually attach an oligonucleotide molecule to this mRNA candidate. So this oligonucleotide capping ensures the stability and efficiency of molecule. So one of the analyses to ensure the quality of mRNA-based vaccine is to do this oligonucleotide capping analysis. So we can do so, as I mentioned earlier, using this instrument so that you can confirm the capping or uncapping of your mRNA vaccine candidate. You can even confirm whether the capping has gone properly or not. And we have done a lot of work on related to COVID-19. So you can just Google COVID-19 resource hub Waters, and you will find our web page with all this information. So in summary, the compact high resolution mass spectrometer we call as Acuity RDA detector is very powerful solution which can simplify the usage of high resolution mass spectrometer for everybody, a chromatographer, scientist, or anyone without having any mass spec experience. The streamlined workflow and the smart features that we have implemented in the solution can help you generate your data very quickly and efficiently right from injection. So it's again designed to allow everyone to use high resolution mass spec data for your analysis. So thank you for your time, and we will value your feedback. So in case you need any material or more, just help us fill up this this survey by using this QR code here, and I will be happy to answer some quick questions that you might have. Okay, thank you.
Okay, thank you, Dhaval, for your presentation. Because the time is limited, we have only 2 minutes. I think there is no question in the chat box. But if the participant that will have some question, you can, Bapak Ibu dapat, karena waktunya terbatas, Bapak Ibu, jika ada pertanyaan, mohon dapat WA ke saya. Saya sudah tulis di kolom chat box atau email ke saya. Nanti saya akan coba, misalnya ada pertanyaan, akan saya coba jawab atau saya diskusikan dengan Mr. Dhaval. Dan untuk acara ini kita akan berikan e-sertifikat, juga materi akan kita share dengan syarat Bapak Ibu mohon dapat mengisi survei yang sudah saya share di chat box ya Bapak Ibu, linknya ada, dan tadi QR code-nya sudah di-share oleh Mr. Dhaval.
Yeah. So I see some questions just sent to me privately. So okay, maybe I'll answer one or two questions. So
okay,
maybe one of the question is whether it can acquire the MSMS data or not. So
mungkin Bapak Ibu intinya.
As I mentioned earlier, the instrument is capable of generating the fragmentation data. So even we have this data independent acquisition mode, where in a single analysis, user can generate a full scan MS as well as full scan MSMS data for your molecule. So you get accurate mass for your parent ion and precursor ion. And using this data, we can do a lot of unknown identification as well.
Iya, Bapak Ibu, intinya alat ini adalah inovasi dari Waters. Alat ini adalah powerful, sifatnya high resolution MS, bisa mendeteksi unknown atau identifikasi unknown unknown compound, baik itu dari small molecule maupun sampai large molecule. Baik itu bisa digunakan atau dioperasionalkan oleh user yang memang mungkin belum terlalu experience di bidang MS, karena alat ini lebih simple, lebih compact dibandingkan high resolution yang lain. Seperti itu. Jika ada yang lebih ingin ditanyakan, nanti mungkin bisa kontak kami di nomor yang saya sampaikan di chat box atau langsung ke Mr. Dhaval.
Yeah. So then other question was about the performance of the system and the mass range. So again, I mentioned in my presentation slide that the instrument can achieve up to 7,000 mass range, so which is more than enough to analyze your small molecule as well as biologics drugs. And although it's small, it has more than 10,000 mass resolution, and with a very good mass accuracy performance that you get consistently from the system. So that's why it can cover all application ranging from natural products profiling, food component analysis, food contaminant, pharmaceutical impurity, and confirming the identity of drugs. And using the biopharma workflows that we have, you can analyze your antibodies, recombinant proteins, oligonucleotides, peptides, as well as your vaccine candidates, as I mentioned in my slides briefly. Yeah.
Ya, aplikasinya luas ya, baik itu di pharma, food maupun biosimilar. Di food mungkin untuk identifikasi sinyal-sinyal impurity maupun residu-residu, kemudian di pharma juga impurities, itu untuk small molecule ya berarti. Dan untuk biopharma bisa untuk vaksin, protein rekombinan maupun oligonucleotida. Alatnya kita namakan Bioaccord. Itu sudah ada beberapa di beberapa negara diinstal di perusahaan biofarma, perusahaan farma maupun food.
Yeah. Okay, maybe this the last question about the libraries. So someone asked if we have dedicated libraries. So usually the libraries are required for small molecule work. So we have libraries for natural products, traditional medicine. We have library for food contaminants. We have library for drugs and the metabolites. And we have library for other molecules, such as if you're looking at excitable leachables. And the software also has a lot of tools if you are doing some impurity or transformation studies to give you an idea how the particular molecule may be transformed. So we have all the libraries available to do the screening experiment. And for biologics, you only need library for Glycan analysis. So I think Waters is only commercial vendor who has their own Glycan library. So if you're doing the Glycan studies, we have libraries using different labeling reagents that you can use it for your studies. Yeah, thank you again for the question. Thank you.
Oke, untuk library, library alat ini cukup luas, mulai dari food kontaminan, drug screening, kemudian natural product, biosimilar, glycan analysis yang sudah dikembangkan oleh Waters. Oke.
Thank you. I think
thank you, Dhaval, for your time. Bapak Ibu, terima kasih atas waktu. Nanti kami akan share materinya, kemudian kami akan memberikan sertifikat juga materi, tapi mohon sebelumnya untuk mengisi survei yang sudah kami berikan linknya maupun QR code-nya. Terima kasih. Oke. Assalamualaikum warahmatullahi wabarakatuh.